Okay, this lecture is on the thyroid gland and the formation of T3 and T4. You'll notice in the bottom left-hand corner, I have added some YouTube sources there for you. Do not be afraid to use those. There's some very good information. I use them myself, and I'll oftentimes emulate a lot of that information and draw it out, adding to or changing it to meet our lecture, and then to add some clinical information into it. Don't be afraid to use those. AIM, MDS, Osmosis, Armando Hasudagan, and handwritten tutorial. Armando's are excellent. I use a lot of his information, and so I would encourage you to look at those as well, and it might help clarify it even better than I can. So don't be afraid to use those. On this lecture today, though, this is a slide or a picture of what the thyroid gland looks like, and we're looking at it from a cellular level. And you'll notice the structuring to this is that they're made in a halo type structure, so that you've got a group of cells that are lining around the outside of these structures, which will form a center. That center is a colloid center. And then you'll have individual cells to the outside. You'll also note that you'll see there is a clear cell in part of these structures, and that's the C cell that produces calcitonin. We're going to come back to that in a later lecture. These other cells that you see listed here, and I'm circling around now, those cells that you see are what are called follicular cells, and they're the thyroid follicle cells, and the center of this is the colloid. Now if we proceed here, you will see that I've drawn this out in a structure or format to give you a little better identification of those cells. Again, each of these individual groups of these has about 20 to 40 follicle cells surrounding this center, and this center is called a colloid because colloid means glue-like, and that's what it is. It's a thick, viscous center. And then you have individual follicular cells surrounding it. And then, of course, bathing each of these is blood vessels. Now, if we take a section of this, let's just say we're going to pull out this section, and we'll pull it over here and magnify it so that we can take a look at what's going on within this structure of things. If we look at this again is just magnified so you can see these thyroid follicular cells. Inside we have our colloid, and then of course our thyroid follicular cells, and then we have our blood vessel that is bathing these. Now inside the blood vessel, of course, we're going to have things like some sodium, we're going to have iodine, and we're going to have potassium, you're going to have chloride, you're going to have more iodine, and all these structures that are listed inside this cell, that are inside the blood rather, that will be transported in. Now inside the follicular cell, if you'll remember, inside of every cell, if you remember your cellular stuff, you have this stuff that is known as endoplasmic reticulum. And it synthesizes protein, puts it in our Golgi apparatus, and then would transport that into the cell. And this is thyroglobulin. Thyroglobulin is a large protein molecule, and it has these tyrosine chains attached to them. So this thing is a long molecule, but it'll have these tyrosine chains that are attached to them. And tyrosine, that's T-R-Y. Tyrosine. These tyrosine chains that will be attached to this thyroglobulin. And there will be many of these. Okay, so then we have our thyroglobulin that's brought in, is produced by the endoplasmic reticulum inside the follicle cell, and these thyroglobulins are large protein chains that have tyrosine attached to them. Okay, now what will happen then is we have these transporters on these cells. This is known as an NIS transporter. And it is going to transport sodium and iodide into the cell. Now remember, this iodide that comes in is iodide, not iodine. It will come in the cell. Now this, we have another transporter here that will transport this iodide then on into the cell, and we'll exchange it for chlorine. The sodium will also be transported back out of the cell, back into the blood, so it saves the sodium. The iodine then comes into the colloid. Inside of the colloid we have peroxidase, which converts iodide to iodine. Okay? And then once the iodine is formed, it then combined with my tyrosine. Okay? And it can do it in many ways. I can have iodine that form here. I can have an iodine that forms here and here, and an iodine here and here. I'm doing this just for simplification because this can do it in multiple places. But what I want you to understand is that if one iodine forms on here, it is known as MIT, which stands for monoiodide iodothyrazine. And if two form on here, is diiodothyrazine. So if I have one iodine on, it is monoiodothyrazine. Two is diiodothyrazine. Once this has happened, what you can see take place then is they combine together through ester bonds, and I'll show you that just a second here. Now our monoiodothyrazine, diiodothyrazine can form ester bonds, and if they do, they will combine together. And if you have a diiodothyrazine that forms with an ester bond that occurs when this knocks off this hydrogen and then forms this bond, so that we would have, you got an oxygen here, and then this one comes down and combines, you would have this makeup then that would occur. Because this is a dye, it had two iodines on it, comes down and forms with this one that has one iodine on it, and now that is T3-T4-Formation. If, say, this D2 or DIT forms with this one, we again would knock off our hydrogen here, okay, and this would become just an oxygen. Then it form here, and we would have T4, thyroxine. Okay? So what will happen within those is you've got this whole thyroglobulin chain, you have these tyrosine molecules on it, the iodine then binds with it, it'll bind with it on one site, it's called a monoiodothyrazine. If it bonds with it on two sites, it's a diiodothyrazine. Through ester bonding, these MITs, DITs can form together. If a DIT binds with an MIT, that would be three iodines formed there to form triiodothyrazine, triiodothyroxine. If two of the DITs form together, that gives you four iodines, and that becomes T4, or thyroxine. Realize this is just a small sample of what we're talking about, because these things are very, very long chains, and you will have multiples of this taking place. So within some of these thyroglobulins, you may have an MIT, a DIT, a T3, and T4s, all within multiple places in these chains. So it's not just one T3 or one T4 or one DIT or MIT that forms out of this thyroglobulin. We can have multiple of those within a thyroglobulin. Okay, now, if we move on over and look at this from the standpoint of our whole makeup here, we will see that inside of the colloid, then we have the thyroglobulin that can have, within that, we can have an MIT, which is one iodine. We can have a DIT, which is two iodines. We can have our T3, which has three iodines formed because of the bonding between a MIT and DIT. And then we can have a T4, which is a bonding of two DITs together. So that forms this, all of the structure. Like I said, there'll be multiples of those. But as large as that molecule is, that's not going to be able to get out of the cell and into the bloodstream. And so what happens is when it gets into, goes back in, this is our old friend, the follicular cell over here. And what's going to happen is this is going to go into the follicular cell. And when it does that, there is inside the follicular cell, there are lysosomes. These lysosomes then will take this in and they'll break off and separate T3 and T4 and transport it into the bloodstream. The MIT and DIT are then reabsorbed and put back into the colloid. The T3 and T4 then can go into the bloodstream, and remember, they are amines, but they are lipid soluble. Therefore, they have to bind with certain protein molecules to be able to be transported. And there are certain thyroid binding proteins that they'll bind to. So when the T3 and T4, which now are separated, so they can go in individually, do have to bind with a protein to be able to be transported. Okay, and that gets you the makeup and the formation of the T3 and T4. And then the rest of the lecture will tell you about how that's going to be utilized in to increase metabolism and in deficiencies and decreasing metabolism. Hope this helps. Again, refer to some of the other YouTube sources and give you further clarification. God bless you.